Hello, welcome to my talk. I'm Priyal De Souza, and I'm going to talk about clinical genomics. The subtitle of my talk is The Basics and an Up-to-Date Technological Perspective. So what is clinical genomics? Well, genomics is a bottom-up approach that uses the DNA sequences to identify genetic changes resulting in molecular mechanisms likely to cause disease. It's become a more frequent way of diagnosing many inherited diseases, cancers, and it's really the future. So in this slide, I'm going to talk about several common types of genomic DNA analysis, starting with the historically the oldest invention around about the 1980s, where clinicians used single gene amplification sequence and sequencing the mutation and variation to detect DNA damage that results in a disease. But that was single genes. That process sort of changed with the requirement of a lot of diseases being caused by multigenic mutations. In other words, mutations in more than one gene. And since the beginning of the century, there have been new inventions like microarrays, which use DNA hybridization panels. And more recently, in the last decade, the next generation sequencing based inventions have come on board, namely the whole genome sequencing, exome sequencing, and ChIP-seq and methylome sequencing protocols. I want to emphasize all these four or five methods I'm talking about are really used in the research and development side of clinical diagnostics. So it's sort of a preliminary analysis is done to build tests which are carried out in the clinics. And this last method I mentioned, targeted enriched next generation sequencing panels, is actually found its place much more commonly now and is becoming common use in clinical diagnostics labs. It can also be used in R&D diagnostics, but I'm going to talk about the clinical diagnostic applications in the next slides. So targeted enriched next generation sequencing panels, they can be bought commercially from biotechnology companies, diagnostics companies that sell an off-the-shelf end-to-end solution, starting providing with the preparation and the reagents to the machines and also the computing algorithms and the hardware that does all the analysis. These panels can be disease specific, as the examples I'll give you in the following slide. The great thing about these disease specific panels are they're all pre-validated. There's no further validation that you need to do, and they fall in. They meet regulatory and accreditation requirements, which are prerequisite in clinical pathology labs. So what is an NGS panel? Well, basically an NGS panel is a series of regions, could be in a gene adjoining each other, or it could be spread through different genes on the same chromosome, or it could also be spread right through different chromosomes. So they don't need to be grouped together in one place. So it could be, as I said, spread through the whole chromosomes. And the basis of one of these regions here in this particular cartoon shows that they actually contain DNA sequence regions which contain previously known mutations, or called mutational hotspots. And so one of these panels consists of thousands and thousands of these mutational hotspot regions. And these regions can be amplified pretty much simultaneously in one single tube. So thousands of regions corresponding to 20 or 30 genes can be amplified at the same time. In the worst case scenario, they might need to be amplified in 2 separate tubes, but it's a single tube amplification system. So basically, this gives you an example of NGS panels. For example, there's a bladder cancer panel here for urethral carcinoma, one for renal cancer carcinomas. Then there's for BRCA, there's breast cancer, or you can get liver cancer. These are only 4 examples. There's many other panels here I don't have time to talk about. So what's actually involved in getting the analysis? Well, you start off by making the DNA from a blood or a tumor sample, and then you prepare the DNA library. Basically follow this workflow here. Library preparation, template preparation, run the sequence on the sequencing machine, and end up getting a data which can be analyzed. So what actually happens with this data? Well, there's clinical interpretation and diagnosis, and that involves the clinician normally gets a report and asks the pathologist to review the automated report that's generated and suggest a diagnosis according to the mutation data produced. That's it. Thank you.